This is part 2 of the Modern Biology 10.1 or 10.3? 10.1 reading, and yeah, we are just talking about replication of DNA, or of the double helix. And if you haven't already seen the first part, I would go see that, because yeah, this is in the middle of the chapter. Alright, so continuing, the complementary nucleotide chains in DNA model led to suggestions of how DNA might copy itself. The ability of DNA to make exact copies of itself is important because, in most cases, cells that divide must pass exact copies of their DNA to offspring cells. So, yeah, we were just talking about how a helix could split down the middle, and that's what they're saying right here. See, I haven't read this chapter yet, but we've been learning in class, so, and the test is tomorrow, so. Replication of DNA. The process of copying DNA in a cell is called replication. During replication, the two nucleotide chains separate by unwinding, and each chain serves as a template for a new nucleotide chain. DNA replication is illustrated in figure 10.5. So I'm going to show that to you real quick. Now, I really don't understand this. Actually, part A shows the replication fork, and then part B shows in the middle of the separation, and part C shows the new chain and the old chain. So if you have this book, you can be looking along with it, or there might be an online version. So, yeah, DNA replication is illustrated in figure 10.5. The first step is the separation of the nucleotide chains. The point at which the two chains separate is called the replication fork. The chains are separated by enzymes called helicases. Now, when I was memorizing the vocab for this chapter, I thought helicases, hell, so hell is you're separated from the person you love. I mean, that's kind of sappy, but it helped me when I was learning in the test. So helicases is separated, and polymerasis, I'm probably not pronouncing it right, but polymerasis is when the separate chains are binded together. So I thought Polly Pocket. When I was little, I used to love it, and I just thought poly would be a good positive thing, and helicases would be negative. So heli separated, polys coming together. You probably can think of another way to put that, but okay. As the helicase enzymes move along the DNA molecule, they break hydrogen bonds between the complementary bases, and the chains separate. As figure 10.5 shows, enzymes called DNA polymerases bind to the separate chains of DNA. So, I don't know if you can see. Okay. Well, the purple there are the helicases, and as you can see in the first one, it's separating the chain, and the second one is still going up and separating, but the polymerases are finding pairs for those separate chains. And in the last one, you can see that they're completely chained together. So if you look, this is a separate one, and then these are the individual strands. Nucleotides are joined together to the new chains by covalent bonds between deoxyribose sugars and phosphate groups. They are joined together to their original nucleotide chain by hydrogen bonds. The complementary nature of the two chains of DNA is the foundation for the accurate DNA replication. Suppose that the sequence of nucleotides in one chain of the original DNA molecule is ATTCCG, which would be adenine, thymine, thymine, cytosine, adenine, guanine. So those are all the different pairs, and that would be one chain of the DNA. DNA polymerasis is when it comes together, would produce a new nucleotide chain, or the sequence of TAAGGC. Now, if you think of that, ATTCCG and TAACC or GGC, T would bond with A would bond with T, A would bond with T, G with C, G with C, and C with G. So it sounds confusing, but it makes sense that the groups, if there's a long chain of these, then adenine, thymine, guanine, cytosine, they all bond together, so they're all going to be in this order and bond together. DNA replication does not begin at one end of the molecule and proceed to the other. Rather, DNA polymerasis begins replication simultaneously at many points along the separated nucleotide chains. Replication occurring simultaneously at different sites permits faster DNA replication. For example, replication is initiated simultaneously at about 6,000 sites in a fruit fly DNA. So this is very, very, very complicated, and all of this crap is happening all at the same time. And we're just looking at a really tiny part of it. Like, we're looking at a microscopic part of the DNA chain, and there's so much complex things going on in this. When replication is completed, two new exact copies of the original DNA molecule are produced, and the cell is ready to undergo cell division. Each new DNA molecule consists of one new nucleotide chain, joined by hydrogen bonds to a nucleotide chain from the original DNA molecule. So, they each form a new molecule, and each one has the new chain that it attracted from the template that we were talking about before. So ATTCCG has a new component of TAAGGC, and they are paired together, and they make up the original molecule. So the molecule split, they have two separate chains, and then the chains they attract the exact same components they had before, so you have two separate, complete, exact copies, so it's replication. Accuracy and repair. The process of DNA replication occurs with a high degree of accuracy, about one error in every 10,000 paired nucleotides. So if you're thinking about these millions and millions of things happening at the same time, all of these things are happening, and every one pair gets screwed up in every 10,000 pairs. So these are very, very, very small things, and they make up a huge part of your genetic code, and they are like building blocks. I know they said the cell is the building block, but DNA is really the building block. So, anyway, if you think of how many cells we have, these are making up, like, it's very, very complex. However, a change in one of the nucleotide sequence at even one location, called a mutation, may have serious effects in new cells. A combination of DNA proofreading and repair processes help keep the error rate to 1 error per 1 billion nucleotides. So, in every nucleotides are the adenine, thymine, guanine, cytosine. So these have to go through and be synthesized in red, and there's only one screw up in 1 billion nucleotides. So that is a very high accuracy. The number of errors and mutations in DNA replications is reduced as enzymes proofread DNA. So it's like if you go through an entire huge book and you have to write this entire book and you don't go back and look at it, but you have an editor, and the editor goes back and looks through it and makes sure that everything's right. So these enzymes are kind of like the editor of the book, and the process of DNA replication is the writing of the book. So it's like you have this thing going through and you make sure you completely do the base of it, and then you have the enzyme going by and making sure everything is right. So that's why there's such a low amount of error. Most repair enzymes detect errors in the complementary base pair structure of DNA. If non-complementary bases are paired, the abnormal DNA structure can be recognized and repaired by specific enzymes. Although DNA replication and proofreading present many replication errors, some errors do occur. In addition, DNA can be damaged by a variety of agents, including chemicals and ultraviolet radiation from the sun. So everything could be good, and then you yourself can kind of screw it up, like staying outside of the sun and like slathering yourself with tanning oil could screw up and mess this stuff up and could possibly even cause cancer. Because cells are continuously proofread and repairing their DNA from the stupid stuff we do, the number of mutations is reduced. Okay, so that's all for the first part, and I probably went over this time limit, but I hope not. I hope this helped you. It definitely helped me, because I didn't even know I knew half of this stuff. So yeah, thanks.